It's late. You awake from a restless sleep. The chief. There's a new case, and it's a bad one. In a haze, you stumble into your clothes and out the door. You're still half asleep when you see it rear up out of the shadows. The old Miller place. Nobody's lived there since... well, nobody's lived there for a long time. Your partner was supposed to meet you here, but it looks like you're all alone. Something smells funny about this, but you decide to go in anyway, against the warning tightening around your heart. As you approach, the door creaks open on its own. What the... You shoulder through and down a long hallway. A musty smell pushes its way into your nostrils. Shadows dart at the edge of your vision. Was that a... No. No. Get a grip, Jenkins. It's nothing. You come to the end of the hall. A doorway. Your heart pounds in your ears. Your whole body is screaming at you to leave, to go away, to get out. But despite all that, you can't go. There's something driving you forward. Something stronger than fear. You step through. Blood. Blood everywhere. Christ.
Hi everybody, it's time to talk about blood. We're going to be looking today at an overview of different characteristics that blood has and learning how we can use it in forensic science to help solve crimes. But before we can do that, we've got to set the stage. So, imagine you've seen some blood at a crime scene. You might have a couple of questions about it that you would need to answer. First of all, is it actually blood? Some things look like blood, but aren't. Secondly, if it is blood, is it human blood? And thirdly, if it is human blood, whose blood is it? So let's take a look at these questions in order, and let's also uncover the context, the additional information we'll need to understand the answers to these questions. So, first question is it blood? All right, so a few things to know about blood. First of all, we've got a lot of blood in our bodies, about 5 liters, or about 1.3 gallons. That may not sound like a lot, but believe me, it is. Or as Lady Macbeth would put it, yet who would have thought the old man to have had so much blood in him? It's surprising. What does blood do for us? Well, its primary purpose probably is to just move stuff around the body, nutrients, wastes, gases that are useful. But it also helps to protect the body from invaders, bacteria, viruses, parasites that might want to invade and make use of these resources you've got around here. And the final thing that it's really good at is stopping its own loss. Blood tends to clot and prevent your dying of extreme blood loss. There are some risks associated with having a circulatory system with fluid flowing through it. For example, you can lose that fluid through massive bleeding or hemorrhage. We can die through total exsanguination, which is the removal of all of our blood. We can also get blood clots that form inside our bodies when we're not actually bleeding, and these clots can travel to our brain, causing a stroke, or lodge in the arteries supplying our heart muscle, causing a heart attack. Some additional information. Let's dig a little deeper. So let's talk about this function of transport. So red blood cells are the cells that are responsible for carrying gases around the bloodstream. And in particular, we're interested in carbon dioxide, which is a waste product of metabolism, produced as we burn, so to speak, molecules for energy, and oxygen, which is something we need in order to make use of our food. The red blood cells are able to carry these gases because they contain the protein hemoglobin, which can bind chemically to carbon dioxide and oxygen. The blood also transports wastes to the liver or the kidneys, where the liver and kidneys can either remove these from the bloodstream or change them into something that is less toxic. The blood is also responsible for carrying nutrients from the digestive tract, where the food comes into our body, or storage areas where we store it. For example, glucose is stored in the muscle tissue or in the liver. Finally, the blood is responsible for transporting hormones all around the body. So if a hormone is secreted, for example, in your brain, that's job is to help you hold on to water because you're running out of water, then that'll travel through the blood down to your kidneys where it will have its effect. And also, the blood is responsible for transporting heat around the body. So if you're too hot, for example, then the blood will carry heat from your warm core out to your skin, where that heat can radiate out into the environment. The second role of the the blood is to be a transportation network for the immune system to help to protect you. So we have immune cells in our blood called white blood cells generally that help to destroy pathogens, viruses, bacteria, parasites, and so on. And the antibodies produced by these immune cells destroy not only pathogens but also some poisons. And we also have platelets in our blood, which are principally responsible for stopping the blood from flowing locally when we do get cut, and thereby preventing a lot of blood loss. Okay, so here is an overview of the composition of blood in the body. So you can see that whole blood makes up about 8%, we're looking at the leftmost column now, about 8% of your total body weight. Of that 8%, about 55% is blood plasma, so that's the fluid with stuff dissolved in it, and 45% are what we call formed elements or cells. These are the cells. So let's look at the cells on the bottom here. Of the cells, we have in every microliter, and there's 1 million microliters in a liter, so it's these numbers times 5 million to get the number in your whole body. For every microliter, hundreds of thousands of platelets, thousands of white blood cells, and millions of red blood cells. So your blood is really densely packed with lots of really useful stuff. It's not just a red fluid, it's a dense solution of all kinds of useful components. And we won't trouble ourselves with going into detail on what the individual white blood cells are called or what they do. Up top, you can see the blood plasma is divided into mostly water, but also other solutes, electrolytes, nutrients, gases, things like that, and then a lot of proteins as well. And of these proteins, we're going to be particularly interested in one category. So the bits in the composition of blood that we're going to be most interested in as we go forward are the red blood cells and the globulins, which are the category of proteins that includes the antibodies, which have really important relevance to the immune system and also to the precipitin testing we're about to look at and the blood typing we're about to look at. So let's dig in. Your first question was, is it blood? And so the Kastle-Meyer test is a chemical method for determining whether something really is blood. And what it tests for is the presence of hemoglobin in a sample. So here's how you do it. First, you take a little cotton swab, you swab at the stain. It can be wet, it can be dry, it doesn't matter. You'll add a drop of ethanol to the swab with the sample on it, and this will burst the cells open. You might recall that cells have a cell membrane or plasma membrane, which is made of phospholipids. These lipids are soluble in ethanol, meaning they'll dissolve in it. So if the boundary of the cell dissolves away, then the rest of the cell just bursts open. Once the cells have burst, we're going to add a drop of phenolphthalein. This is a pH indicator. If you've worked with any pH indicator, you've probably seen phenolphthalein. It's the one that turns pink when the pH changes. You've probably used it for titrations. And then finally, you're going to add a drop of hydrogen peroxide. And here's the results. If it is blood, the end of the swab will turn pink, or really it's more like sort of a magenta color. Well, you'll see it in person. If it's negative, which is to say it's not blood, then no color forms at all. It'll just stay whatever color it was when you started the test. Now, how does this work? We won't go into a ton of detail on this, but the basics of it is that it's a redox reaction. So the hydrogen peroxide pulls electrons away from the hemoglobin, so the hydrogen peroxide oxidizes the hemoglobin, and then the oxidized hemoglobin will then pull electrons away from the phenolphthalein, oxidizing it, and the oxidized phenolphthalein has a pink color. So it's sort of a chain reaction of redox, which ends up with a pink-colored product. If the hemoglobin's not there, though, then you won't get this pink color. So here are the reagents we'd use. So you can see on the left, it says ETOH, that's some shorthand for ethanol. In the middle, KM stands for the Kastle-Meyer reagent. It's basically just phenolphthalein that's been chemically treated in a certain way. And then on the far right, you've got your hydrogen peroxide, which is the final step. At the bottom of the frame, you can see a positive test result there. It's that pinkish sort of fuchsia magenta color. And then below it, a negative test result, which is just the color of the cotton swab. Really important to keep in mind, you can get false positives with the Kastle-Meyer test. So anything that can oxidize the phenolphthalein can turn the test pink. So there are lots of things other than blood that can do this. That's really important. So we say that the Kastle-Meyer test is a presumptive test for blood. That is to say, if it's not blood, it won't turn pink. But if it turns pink, it could be blood, but it might not be. So if it turns pink and it looks like blood, then you should probably collect the sample and test it further. But if it doesn't turn pink, then maybe it's just like food coloring or ketchup or something, and you don't need to worry about it. So we think it's blood. We take it into the lab. We want to know, is it human blood? We need to know a little more about blood before we can answer this question. Okay, so what we need to know is about antigens on the blood cells. So the surface of cells, all cells really, are covered in different molecules, lots of different types of molecules in great abundance that serve a lot of different functions. And we call these molecules on the surface antigens. And their shapes are unique. So all these surface molecules have specific, unique shapes. These shapes are specific to those molecules in particular. And so the shapes of molecules on the cells of humans differ from the shapes of molecules on the cells of other animals. So human surface molecular shapes are different than dog surface molecular shapes are different than goat surface molecular shapes, and so on. It's species specific. And this is actually really important. This is how the human immune system tells the difference between its own cells, the good guys, the ones we're trying to protect, and those of an invader, like a parasite or a bacterium that is harmful. By the way, most bacteria are not, and the immune system won't go after them. But if they're harmful bacteria, then the immune system will. And they do this by checking these shapes on the surface of the cells. So if there are human shapes present when the immune system checks, then the immune system doesn't attack. Everything's cool. And if non-human shapes are detected, then the immune system goes ahead and tries to destroy the invader. So here's an image of the surface of a human cell. That double layer of red is representing the cell membrane. And you can see all these things protruding on the top side of the red band. These are representing all sorts of surface molecules. And you can see they all have all these different shapes. They're really quite varied. So in addition to antigens on the surface of the cells, we also have what we call antibodies, which are free-floating in the blood. And the way this works, the way we check the shapes, is by using these antibodies, which are sticky proteins in the blood. So antibodies, like antigens, have various unique shapes. Some of them form complementary shapes to the shapes of antigens that are on the surface of cells. So you can imagine maybe this is a antigen shape. The complementary antibody might have a shape that looks like this, and they fit directly together. And so the human body produces many different sticky antibodies with different shapes that match the shapes of molecules found on the surfaces of foreign invaders like parasites, bacteria, and viruses. And when these antibodies stick to the antigens on these invaders, they cause the cells they're sticking to clump together. Basically, the antibodies stick to the antigens and they stick to other antibodies, and so they all sort of stick together and clump up, and we call that agglutination. And you can actually see it, and I'll show you some pictures in just a minute. So here's a cartoon diagram. You can see the virus in the center of the screen, and the antigens are these little blue spheres sticking out from the surface. And then you can see the antibodies in the periphery in a red color, and those are matching the shape of these spheres protruding from the virus. Of course, the shapes are simplified to make it easier to see. Most of the shapes are quite irregular. And here you can see a cartoon diagram of the process of agglutination. In this case, the purple is representing the shapes of the antibodies, and the yellow is representing the antigens on the surface of the green invader cells. And you can see how they clump together here. So, how do we use this information about antigens and antibodies to figure out whether something is human blood? Okay, so follow along with me. Because human blood contains human-specific antigens, which are only found in human blood, we can test for them using specific antibodies that will stick to human antigens only. That's the thing about antibodies. They'll only stick to one antigen. So if you can get antibodies that stick to human blood and human blood only, you can use them to test for human blood. So the blood that we suspect that we took from the crime scene is added to a test tube along with a solution of antibodies that stick to human antigens. If the blood really is human, we'll see agglutination where the two solutions meet, and we'll get a visible solid gelatinous layer where they meet. So you'll have the antibodies down here at the bottom, and you'll have the maybe blood here at the top, and you'll get a gooey layer where agglutination is happening at the interface of the two fluids. If the blood is not human, then we won't see any reaction. So here's what it looks like. You can see the test tube here. On the top is a sample of what we think is human blood. On the bottom is a solution of antibodies which will stick to and cause human blood to clump up. And then in the middle, you're seeing a positive test result. You're seeing this gelatinous layer where the human blood cells are clumping up because of the action of the antibodies. And so this is a positive test that the blood really is human. By the way, you might wonder, why do we need to do this test? Isn't it going to be human blood if it's a murder scene? Well, it might, or it might not be. For example, imagine that you have a person who's a suspect in a murder, and they are an avid hunter, and you find some blood in the trunk of their car, and that could be human blood from the victim, or maybe they shot a deer over the weekend and they were transporting it in their trunk. It's possible. The precipitin test will allow you to distinguish between those two possibilities. All right, so you figured out it's human blood. You figured out that it is blood in the first place. Now you want to know whose blood is it. We need a little more detail to figure that out. What we need to learn about is blood types. So let's start with the ABO blood types, then we'll add another layer of complexity. So in addition to the surface molecules that flag a human cell as human, we have additional markers that are different from person to person and are shared by only a few people. So a person can have a surface molecule we'll call A, a surface molecule we'll call B, neither or both on the surface of their cells. There's lots of variation in the population. So if a person only has molecule A, we say they have blood type A. If they only have molecule B, we say they have blood type B. If they have both A and B, we'll say they have blood type AB. And if they have neither molecule A nor molecule B, we say they have blood type O. This is what we call the ABO blood typing system. There is an additional factor, though, and that's what we call the Rhesus factor, or just the Rh factor. It was first discovered in Rhesus monkeys, hence the name. If a person has the Rh molecule present, we say they have a positive blood type. We add a little plus sign next to their blood type. If they are missing the Rh molecule, then we say they have a negative blood type, a little negative sign next to their blood type. So if you have molecule A and the Rh molecule, then your blood type is A positive or A plus. If you have molecule A and B, but not the Rh factor, then we'd call you blood type AB negative or AB minus. If you had neither A nor B nor the Rh factor, then we call you O negative. So here's the really important part for our purposes, and that's that if a person has a certain antigen, either A or B, the Rh factor, any combination of them, they will not make antibodies against that antigen. How do we know this? Well, if they did, they'd be dead because their blood would be constantly clotting, clumping up, and they wouldn't be able to transport molecules around their blood. They'd be dead in a matter of moments. They'd have sort of like all of a sudden strokes and heart attacks of massive proportions. So this is really important to keep in mind. Okay, so here is a chart of which antigens are present in which blood groups and which antibodies those people will actually make. So if we just take it from left to right, starting with the first column, you can see someone with blood type A, it says group A, will have these A-type antigens on the surface of their cells. They may have in their blood anti-B antibodies, but they won't have any anti-A antibodies. If we now go over to blood type B, we can see there's B-type antigens on the cells. They might produce anti-A antibodies, but they definitely won't produce anti-B antibodies. If you are blood type AB, then you have both A and B antigens. You will produce antibodies to neither A nor B. And if you are group O, then you don't have either of these antigens on the surface of your cells, and you can produce both anti-A and anti-B antibodies. All right. So there's some variation in the different blood types in the population. You can see here from bottom to top, the most common blood types are O positive and A positive, followed by B positive, then O negative, A negative, AB positive, B negative, and AB negative. So there's some chance that a person in the population will have one blood type or another. They're not all equally common. Okay, so we know a little bit about the blood types now. How do we actually test for them? Here's what we do. We take the blood from the crime scene and then we add certain antibodies to it and see whether it clumps up or not. And so if the blood agglutinates when anti-A antibodies are added, the blood type includes A. Could be A, could be AB. If the blood agglutinates when anti-B antibodies are added, remember those are antibodies that stick to the B antigen, then the blood type includes B, could be AB. If it agglutinates when the anti-Rh antibodies are added, then the blood type is positive. So now I want to give you a little question. I'd like you to pause the video and determine the blood type based on this result. And I'll say, if the blood agglutinates when anti-B antibodies are added, but not when anti-A or anti-Rh antibodies are added, what is the blood type? Take a moment. Pause the video. Think about it. I'll wait. Did you figure it out? Yeah. That's right. It's B negative. Good job. All right, so here's what a test result would actually look like for that blood type. You can see the little area for the A test is nice and clear and red. The area for the Rh test is nice and clear and red, but the area for the B test has these fine little chunks, and those are actually chunks of red blood cells all sticking together. So this shows that the B antigen is present, but the A and the Rh are not, which makes this a B negative blood type. All right, so what's the relevance of blood typing to forensics? Well, if people have particular blood types, we can link someone to a blood sample found at a crime scene by seeing if the blood at the scene is the same type as theirs. If the blood type is the same, the blood might be theirs, or it might not be. Remember, there's lots of people with the same blood type. It could be theirs, but it might not be. However, if the blood type is different, the blood cannot be theirs. For the above reasons, blood typing is better for ruling out suspects than it is for incriminating them. So for example, if we find type AB positive blood under the fingernails of a victim, and the key suspect is blood type O negative, well then the victim must have scratched somebody other than the suspect while defending themselves. This doesn't mean the suspect was uninvolved, it just means that blood underneath the fingernails didn't belong to that suspect. If that was the key piece of evidence that we had to link the suspect, then this person probably we can't build a case against them. So blood typing, better at excluding than it is implicating. All right, let's review. So blood is the critical fluid in the body, serves transportation and self-defense roles. Hemoglobin in red blood cells carries oxygen and CO2 throughout the body. Cell surface molecules called antigens help our bodies tell the difference between our own cells and invaders. Think of them as little ID tags on the surfaces of cells. Antibodies bind to antigens in a sort of shape-dependent manner, right? If it's this shape, then the antibody looks like this. If it's this shape, then the antibody to bind would have to look like this, and so forth. And that causes them to clump up. The Kastle-Meyer test turns pink in the presence of blood by reacting with hemoglobin in the blood, in the red blood cells, and it is a quick and cheap, but inconclusive way to test for blood. The Precipitin test tests for the presence of human-specific antigens and is a conclusive test for human blood. A little harder to do, though. And then blood typing tests for the presence of human antigens that vary within the population and is a good way to exclude a person from suspicion. However, it is not conclusive proof that the blood belongs to a specific person. So this has been an overview of blood. I hope that this was interesting to you and helpful, and we'll see you next time. Thanks.